Okay, we can all see that, right? Yeah.
Yep, I can see that fine. Yeah.
Perfect. So I'm Sarah Raffan. I am a postdoc at Rothamsted Research, and I'm going to tell you about the work that we're doing on genome editing for low acrylamide wheat. So what is acrylamide, and why do we care about it? So acrylamide is a food processing contaminant that was unexpectedly discovered in many foodstuffs back in 2002. Now this caused a bit of a stir because acrylamide is a pretty nasty chemical. Some of you might be familiar with it from like Page in the lab, polyacrylamide gel electrophoresis, but it also has widespread industrial uses. It causes cancer in rodents and it's regarded as a probable human carcinogen. It also has developmental and neurotoxic effects, as well as affecting fertility at high doses. So major contributors to dietary acrylamide intake in Europe are fried, baked and roasted potato and cereal products, as well as coffee. And many of our favorite products are affected. The beige diet is particularly affected, which is in fact my diet, as well as coffee is obviously quite affected. So wheat represents one of the major sources of dietary acrylamide intake, and products such as biscuits, breakfast cereals, bread, toast more so, cake, that kind of thing. Our favorite products are all affected by high acrylamide concentrations. So this is based upon data collected by the European Food Safety Authority in 2011, showing the contribution of different food groups to dietary acrylamide intake from food. And wheat contributes to all the groups that are highlighted there in red. This is quite a lot, and it's about 32% of our dietary acrylamide intake from food comes from wheat-based products, with bread alone contributing about 15%. Obviously, this is a bit of a concern, so we have regulations on this. The European Food Safety Authority's expert panel on contaminants in the food chain, CONTAM, issued a report on the risk of acrylamide back in 2015. And this report stated that the margins of exposure for acrylamide indicated a concern for neoplastic effects. So basically, the amount of acrylamide that we're eating was a concern for cancer-causing effects. As a result of this, the European Commission reviewed its risk management measures and issued these new proposals back in June 2017, which came into force in April 2018. And this is Commission Regulation 217-2158. Now, this regulation includes things like businesses now need to monitor the levels of acrylamide in their food products, as well as keep a record of all of the mitigation methods that they use, so to basically record what they're doing to lower acrylamide levels. And we had indicative values on the levels of acrylamide that you could like in food, and these were rebranded as benchmark levels, and the levels were reduced in many cases. And now, if a product's found to exceed the benchmark level, then the codes practice, the mitigation methods need to be investigated to show that they have done everything possible to lower the acrylamide in that product. As I mentioned, codes of practice, these are now compulsory mitigation methods that businesses have to follow. And these are based upon a lot of the methods that they'd already compiled from the Food Drink Europe Acrylamide Toolbox, which is where industry came together to kind of share their acrylamide mitigation methods. And those are now part of compulsory codes of practice in this regulation. The regulation also went as far as to threaten maximum levels. So these are levels above which it would be illegal to sell a product for sectors that are not showing sufficient progress. And it's already started moving towards this and strengthening the regulations further. There's a bit of uncertainty about this. We don't really know what they mean by sufficient progress. And we also don't know how this will be affected by Brexit and whether or not this will follow into the UK the same way that Europe's going. But it is a bit of an issue in food, right? Acrylamide is an issue in food. But where does it come from? Like I said, it was a food processing contaminant, and it actually forms during the Maillard reaction from the reaction of free asparagine, so non-protein asparagine, and reducing sugars. The Maillard reaction might be familiar to anyone that's a bit of a cook. It's actually not just one reaction, it's a complex series of non-enzymatic reactions between free amino acids and sugars during cooking that gives fried, baked, roasted, cooked food products their characteristics, textures, flavors, smells, and aromas, basically. Basically everything that makes like toast toasty or roast potatoes, the colors, the textures, the smells that you associate, that all forms during the Maillard reaction. And acrylamide forms during this reaction too when asparagine participates in it. Now, this means that acrylamide formation is linked to colour and flavour development, and this forms the basis of the UK's Go for Gold campaign that was issued by the Food Standards Agency in 2017. And this encouraged consumers to cook their foods to a light brown colour, so to stop like overcooking toast or chips or roast potatoes. So the reaction of free asparagine with reducing sugars leads to the formation of acrylamide, and is actually the predominant route for acrylamide formation during cooking. So as free asparagine and reducing sugars can therefore be seen as the precursors to acrylamide. And in cereal products, it's actually the free asparagine concentration that determines the acrylamide formation. So the more free asparagine you have in the raw food product, the more acrylamide that you'll get in the cooked food product. And actually, considering the levels of asparagine in the raw material is part of that regulation I talked about earlier, Regulation 217-2158. And it's now part of it compulsory that businesses do consider the free asparagine concentrations of the raw material that they're using. Asparagine itself is synthesized via the ATP dependent transfer of an amino group from glutamine to aspartate, releasing glutamate as a byproduct. And this reaction is catalyzed by a family of enzymes called the asparagine synthetases. So this model here shows the asparagine biosynthesis, and the metabolites are shown in circles with the reactions shown as squares. And the key thing is that this reaction is underpinned by the asparagine synthetases. You've got asparagine synthetase binding glutamine, taking the amino group, releasing glutamate, binding aspartate, and transferring the amino group to aspartate, creating asparagine. So this reaction is underpinned by the asparagine synthetases. So we know that they're the key players, but what do we know about them? So we know that there are 5 asparagine synthetases in wheat, asparagine synthetase 1 to 4, with a double copy of ASN3. And this figure here shows that intron-extron structure, so it's a divergence between the genes based upon the Chinese spring reference sequence. And interestingly, some varieties lack a B gene inversion of ASN2, such as Chinese spring, which is shown here. There's no B gene inversion of ASN2. Fortunately, these asparagine synthetase genes are differentially expressed. So this here shows this, the expression of ASM1, 2, and ASM3 combined. And it's clear that ASN2 is the most highly expressed in the embryo and the endosperm, so in the grain. And if you look at it, it's kind of only really expressed in the grain, which would make it a really good target for genetic interventions if you wanted to reduce asparagine accumulation solely in the grain without affecting asparagine accumulation throughout the rest of the plant, as asparagine is an amino acid and could be vital for life, is vital for life. So this is an RNA-seq data set showing gene expression in wheat grain at high and low sulfur. So sulfur availability is one of the environmental effects which is known to affect free asparagine accumulation in wheat grain, with asparagine increasing under sulfur deprivation. Overall, there was a much higher expression of the ASN genes in the embryo than the endosperm, so you can see the difference in the axes there. But out of all of these genes, ASN2 is still the most highly expressed, confirming that it's probably the best target for genetic intervention. No B genome was detected in this data, and actually the A genome was much more highly expressed than the D genome, indicating that targeting the A genome alone could be enough to result in a step change in asparagine accumulation in the grain. This would be good just in case there was a phenotypic effect of reducing asparagine accumulation. So maybe we could do a step change and have less of a detrimental effect if there was one. So I've mentioned the B genome a lot. This is work done by Joe Oddy, who's a PhD student in the lab, and he investigated this B genome deletion. So he's actually screened a number of wheat varieties now and found that the deletion is pretty common in UK wheat varieties. It is also present in many of the related wild and cultivated species. It's about a 13,000 base pair deletion. It's responsible for the loss of ASN2 on 3B. And Joe also does work on the environmental effects and the G times E effects on asparagine accumulation. So I mentioned sulfur just a second ago, and Joe's investigating that further, as well as the effect of other nutrients on asparagine accumulation. So ASN2 looks like a good target. And we know that natural variation in the ASN2 genes causes variation in acrylamide forming potential. Because I just mentioned Joe was looking at the B genome deletion, and actually the presence of the B genome ASN2 gene is associated with higher free asparagine accumulation. So we know that without the B genome version of ASN2, wheat varieties have a lower acrylamide forming potential. So we know that that variation does underpin acrylamide forming potential. But what if we knocked all of the ASN2 genes out? What would happen then? And this is what we aim to do with CRISPR-Cas9. So we designed 4 guide RNAs to target the first exon of ASN2. And the reason we wanted to target the first exon of ASN2 is that's where components of the glutamine binding domain are, one of the active sites of asparagine biosynthesis, with the other one being the aspartate binding domain. So the residues that are there highlighted in yellow are components of the glutamine binding domain, and they're also kind of shown by sort of purple annotations on the bar at the bottom, spanning the first exon there. So that's what we wanted to target and knock out. And the guide RNAs correspond to conserved regions in the 3 homologs, so the A, B, and D genomes simultaneously. This work was done in condenser, which obviously had to have a B genome, so to target all 3 of those simultaneously. And these guide RNAs, these 4 guide RNAs, were introduced as a single polycystonic gene, tandem mirroring with tRNAs, and this was biologically bombarded alongside a codon-optimized Cas9 gene and a herbicide tolerance gene. And this is the workflow for the genome editing work that we did. So focusing first on the T0. So we did actually 6 separate bombardments, resulting in 92 plants in total. With 77 of these plants, or 84% of these plants, showed Cas9 integration. And we self-pollinated the T0 to generate the T1 seed. We took 14 of these T0 plants forward, mainly on the basis of their health and on seed number. We did attempt to screen them for edits, particularly looking at things like band shifts on the gel and those kind of methods, but it wasn't particularly successful, and so later on we relied on next-gen analysis, which I'll get on to. However, the subsequent analysis of the T1 generation showed that there was likely to be editing in 11 of the 14 T0 plants that we took forward. So focusing on the T1, we had 14 T0 plants we took forward to generate 167 T1 plants. And we subjected these to next generation sequencing to look for edits in them. And there was a lot of editing. Out of the 167, 107 of them showed editing in some forms. So this could be a single edit at one of the guide RNA sites in one allele of like the A genome, for example. However, 55 of them showed simultaneous editing in the A, B, and D genomes, at least heterozygously. And mapping this back, that comes back to 9 of the original 14 T0 plants we took forward. So this is a really high level of editing, with only 22% of the plants appearing wild type, and then there were 13% that we couldn't analyze in sufficient read number. So from this, we picked 7 lines to take forward to the next generation. We picked 4 that appeared to be triple mutants, so triple knockouts. 2 that appeared to be A genome knockouts, because as we said, the A genome was shown to be much more highly expressed, so they might result in a step change without any potential, or without perhaps the same detrimental phenotype if there was one. And we also took 1 wild type like plant, which had gone through all the process but didn't show any editing. And we grew these plants up and subjected them to next-gen analysis as well. And this was analysis was run on a lot fewer plants and gave us a lot more information actually. So most of the edits that we saw were deletions, although there were some substitutions and a few single base pair insertions. And the edits resulted in what were predicted to encode truncated and dysfunctional proteins, although there were a few that just encoded proteins that just lacked the glutamine binding domain. Interestingly, there were differences between the T1 and T2 next-gen analysis. So in some of the cases, we picked up some wild-type alleles that we didn't see in the T1, in the T2. So one of our triple nulls ended up being just an AD null. But more importantly, perhaps, there was evidence of continued editing, particularly in one line, line 41, which I think, let's see if I can get the pointer up there, corresponds to this set of alleles here, which was originally believed to be an A genome null, but now had editing in all 3 genomes, as well as multiple alleles in the A genome. As we put the lines for stability, this line wasn't taken forward, obviously, but it was just interesting to see continued editing there. So we've shown that we've got really good editing, really high editing efficiency, but do the plants show a phenotype? I mean, that's the most important thing, ultimately. So this is the amino acid data of the lines that we're taking forward for further investigation, in both the T2 at the top there and T3 at the bottom. The blue ones correspond to the wild type and wild type like, the red ones are the triple nulls, and the yellow is the A genome mutant. And first thing to say, the asparagine concentrations were generally a lot lower in the T2 than in the T3. If you can note, there's a completely different, like the axes are very different on them, and we think this is maybe due to stress. The triple knockouts were lower in, or significantly lower in asparagine concentration compared with the wild type. In the T3, we didn't analyze the T2 just as there wasn't enough material to do so. But in the T3 generation, asparagine made up nearly 41% of the total amino acid pool in the wild type, compared to 8% in the T2, which is what would be considered normal. So this is why we think perhaps they were stressed. Asparagine making up that higher percentage of the amino acid pool is quite unusual. But it was nearly 41% in wild type in the T3, with line 178, the A genome null, showing 36%, line 23, a triple null, showing 29%, and our lowest line, line 59, showing less than 18% of the amino acid pool being asparagine. And if you remember back to the beginning of the talk, I said a few other amino acids involved in the asparagine biosynthesis pathway, because asparagine is synthesized from transferring amino group from glutamine to aspartate, releasing glutamate. So looking at these amino acids is also particularly interesting in this. So this is just looking at line 59, which is our lowest line in asparagine, and looking at the other key amino acids in the asparagine biosynthesis pathway. So in red, we've got asparagine, yellow, we've got glutamine, blue, we've got glutamate, and green, we've got aspartate. And glutamate and aspartate showed significant differences to the wild type. So significant changes there in the levels of those. Alongside the CRISPR lines, we also generated tilling lines. And these were generated because at the beginning of the project, so this is back about 5 years ago, the regulations of the gene editing were uncertain, and I mean, they're still uncertain. So we utilized a EMS mutagenized population to develop these tilling lines. And we identified stop codons in the 3 homologs A, B, and D. So this population is also in cadenza, so also has a B genome. However, they have been crossed into Claire, which lacks a B genome, so that's something. And they're being crossed at RAGT to generate a triple null. So these haven't been tested for asparagine concentration yet, but I mention them because I'm about to come on to the field trials. Because we know that knocking out ASN2 causes a reduction in asparagine under glass. We don't know how this holds up in the field. And I have alluded to the fact that there are environmental and G times E effects on asparagine accumulation that Joe's looking into. Well, so we're going to currently test these in the field. And this has generated a fair amount of media interest recently. We believe this is Europe's first genome-edited wheat field trial, although it's not its first genome-edited plant field trial, as there was camelina going on before this. But yeah, this field trial is taking place at our GM site. It still has to be currently still considered GM. And these plants do have some transgenes left in them. And we're currently testing 2 different line 23 varieties. So that's a triple knockout, and one of the line 178 varieties. So that's an A genome mutant. And these are being trialled next to the two lines I just talked about, although only in a genome telling mutant, as the triple mutant is still being developed. So they're going to be trialled, well, that was drilled about a month ago now, and that photo was taken yesterday. So they have come up, they are growing, and we just have to see how they do. We're very excited about that. Those of you who might have been paying a lot of attention have realized that I talked about line 59 as being the one that's lowest in asparagine, but it has not gone into the field trial. And that is because it also has an edit in one of the ASM1 genes. So the ASM1 gene is the most similar to the ASN2 genes out of all of the family members. So if editing were to occur, that is where it would, and it did happen in one of the lines in line 59. It has a 37 base pair deletion in the B genome of the ASM1 gene. And this might explain why it's always been slightly lower than its counterparts in asparagine accumulation. This editing only occurred at one of the 4 different editing sites. So I used 4 guide RNAs, and this editing only occurred at one of the sites in one of the genomes. But this was the site that was most similar to ASN2 site, so it's the site in ASN2. There were 2 mismatches there to the guide RNA, but there was an intact PAM sequence. So this indicates that 2 mismatches might not be enough to prevent off-target editing. Although I should remind you that we had a really high editing efficiency. So it might be a product of that. Anecdotally, other experiments in the lab have shown that 1 mismatch is enough to stop editing. So it could be a product of the editing efficiency here, that 2 mismatches was just not enough. So in conclusion, we did see a really high editing efficiency from our guide RNAs, and this resulted in dysfunctional ASN2 proteins and reduced free asparagine concentration in wheat grain under glass. However, we did also see some off-target editing. So yeah, I have to thank quite a lot of people for helping me with this. Nigel Halford is my supervisor. I'm line manager now. Joe Oddie, a PhD student in the lab, who is working on the BGM deletion as well as environmental and G-time-C effects. Serial transformations for all their work, and the Glasshouse staff, as well as resources from Alison Hutley and Andy Phillips. Lots of thanks go to University of Bristol for the sequencing and bioinformatics that they've done, and Calculus Analytics for the amino acid testing, as well as all the project partners. And this is the current wheat acrylamide team, low acrylamide wheat team. And you might guess I'm on the right there. It's Joe Oddie at the back, the very back of the glasshouse there, the PhD student I talked about, and Nigel Halford on the left there.
Great, thank you very much, Sarah. It's very interesting. It's good to see all this gene editing stuff we hear a lot about, that it's actually getting into the field, and to see if it actually makes any effect, if it's going to be the great panacea that we hope, many of us hope it will be. So, okay, so please, if anyone's got any questions for Sarah, please put them in the Q&A or raise a virtual hand. So let me ask, first of all, let me ask a bit of a technical question about those guides. So I know when people started using genome editing, a few years ago now, the advice was to use multiple guides to increase the chance of success. And you did mention about one of the guides just towards the end there, but did you see consistently that one or other or of the guides worked better than the others? Could you predict that from the sequencing that you saw?
Yes, so I'd say 2 of them worked better than the other 4, and 1 of them worked less well, if that makes sense. So okay. Yeah. And one of the ones that resulted in the off-target editing, or off-target on-target editing, depending how you want to look at it, was one of the ones that was was the most efficient guide RNA. It did edit pretty much out of all the plants that were edited, they would have been edited there.
And can you tell what was there a reason for that? Was I assume all the sequences were matched, but I mean, is there anything about that sequence that allowed you to realize why it was edited better?
So when I designed them, we ran them through a couple of different softwares. That one always came out as having high activity, but also looked at the kind of predicted 3D structure of it, and it did seem like the most clear, the most free to interact. Whereas the one that was the least efficient did have a few hairpins in there that might have stopped it interacting as well.
Okay, so question from Chris Walwick here, and this is kind of an interesting question, which which would be important ultimately for your work. So he says, thanks, very interesting talk, Sarah. Is there any information about whether acrylamide contributes directly to the nice flavor of baked or roast food, or is it totally irrelevant for that?
I don't know, but a lot of businesses have to reduce acrylamide forming potential anyway, and there are a lot of mitigation methods that are in place. I mean, flavors of products might have changed, whether or not that's to do with the fact that the cooking has changed other things, like the change in processing has changed other things, or whether or not it's to do with acrylamide. I don't know.
Nice is subjective, of course, but it's all very well if you have something which is better for you but doesn't taste as good, then that will test the public whether they what they really want, I guess. So.
I think I should say that acrylamide probably doesn't have a flavor as far as we're aware. So
Okay, a question from Chris Stevens. He says, great presentation, Sarah. Did you see any immediate phenotypic differences in your gene-edited plants compared to wild type in the glasshouse, I guess?
There was a germination phenotype that pops up every now and again, not always, and it's been seen in other low asparagine lines. So that's something that we do watch out for, and it can be overcome with the exogenous application of asparagine. So if you just spray the germinating seeds with asparagine, that's fine. That does pop up every now and again, but other than that, there's no other phenotype that we've seen under glass so far.
Okay.
The ones in the plant, the ones that we just put in the field germinated just fine, really well, actually.
Excellent. So a question from Abdul Menon, and he asks about off-target effects. So you mentioned about the AS1, but then did you see, when you did next generation sequencing, full genome sequencing, did you see any other off-target effects at all?
We didn't sequence the whole genome, we only sequenced the target sites.
Oh, sorry. Okay.
So no, we didn't see any others in the AS1 genome that you would be the most similar to anything else that you would see. Only saw that one edit that has popped up. So I think that would be the most likely place to go. So
of course. And I kind of a follow-on question from that. You said that one of the lines was showing continual editing. So did you look to see if the Cas9 gene was still in there, or it had been crossed out the other lines? Or?
So the ones that were taken forward based upon having one of the big contributing factors, whether or not they have transgenes left. So we're trying to get rid of those as much as possible. The line that showed continued editing did have Cas9 still integrated, and presumably still active.
Yeah, I see. Yeah, absolutely. And I guess you don't know at the moment. So obviously these field trials are still in a controlled environment. Is it all fenced still?
It's fenced, yeah.
Okay, so we don't know yet. But maybe you can offer an opinion about what you think. So if this goes fantastically well, and there's low acrylamide and they're super healthy new wheat, what do you think you might have to do to have these lines be available to go out with regard to the amount of transgenes which are in the lines and things like that?
We have identified some of the line 59 ones that are transgene free. They're just not in the field trial due to the off-target, on-target edit.
Okay.
And so we'd like to trial those at some point. And we are crossing that with triple nulls to remove the last of the transgenes. However, there are a lot of IP issues on CRISPR, so that's something to consider before we'd ever consider taking the lines forward.
Yeah, of course.